Let's look inside a testis. Like other important organs, the testes are surrounded by a serous membrane, and that membrane includes a parietal layer and a visceral layer with a fluid-filled space in between. The outer layer of the testis is called the tunica vaginalis, and it is this entire layer right here with both a parietal and a visceral layer and a fluid capsule in between. The outer layer helps form the scrotal cavity, and it covers the anterior and lateral faces of the testes. This same structure, the tunica vaginalis, is also present around ovaries. The tunica albuginea, which is a word that means white, is an egg-shaped connective tissue mass that directly covers the testes. It dips down in between lobules of the testes to form septa, similar to that what we saw in the lungs. These septa can create 250 to 300 lobules, and one testis can have 1, 2, or 3 main areas containing lobules. The bulk of the testis is made up of seminiferous tubules, which are very tightly coiled structures where spermatogenesis occurs. That is the process of producing sperm from a stem cell all the way to the formation of the flagella. These tubules, they converge into straight portions that lead to this structure here that is called the rete testis, which then uses the efferent ductules to channel sperm into the epididymis. In histological views, these tubules, they appear as round or oval structures, much like the kidney, and they have a central lumen. Sperm develop from the outer wall of the tubule and move toward the lumen, eventually detaching and moving into the epididymis. Inside each tubule are many different types of cells, and not just developing sperm cells. All of the seminiferous tubules straighten out and converge upon that rete testis. It's here that they merge together and kind of get intentionally clogged, allowing the sperm a little bit more time to move through the structure and a little bit more time to mature. The flagella of the sperm are not fully developed at this point. So these efferent ductules have ciliated epithelium to help with movement, and the rete testis itself then distributes the spermatozoa to the epididymis using those efferent ductules, which are also ciliated, but also have some smooth muscles surrounding them. After traversing the rete testis, spermatozoa move into the epididymis through those efferent ductules and wait until ejaculation or reabsorption. Spermatogenesis is the process of cell division that takes the stem cell and makes a spermatid, which will then mature into a spermatozoon. This is the process that takes place in the seminiferous tubules. Spermatogonia are stem cells that like to do mitosis. One cell replaces the stem cell, and another cell is designated to go forth and do mitosis and become a spermatid. The mitosis of a stem cell happens every 16 days, and it takes about 64 days to divide and mature into a spermatozoon that's able to detach and enter into the lumen of a seminiferous tubule. At any given time, a tubule is hosting 5 different cohorts of developing sperm. Before this lecture, you may have thought that sperm is produced continuously, and it kind of is, but it's more produced in waves as these cohorts mature. The rete testis that clogging mechanism. It's always controlling the movement of these waves into the epididymis in accordance with volume. There are several cells in seminiferous tubules that support the function of making spermatozoa, but they're not directly related to making spermatozoa. There are myoid cells, which are basically smooth muscle cells, and these surround the entire seminiferous tubule. When they contract, they help the movement of the sperm along the seminiferous tubule into the rete testis. There are also interstitial cells, which are also called interstitial cells of Leydig. And it's these interstitial cells that are prompted by FSH to start making testosterone. The last type of cell that we have has a few names. It can be called a nurse cell, a sustentacular cell, or a Sertoli cell. And what it does is it acts like a gelatinous matrix that the developing spermatocytes are embedded in. It's kind of like fruits and gelatin. And these sustentacular or nurse cells, they provide nourishment and structural support for the developing spermatocytes. In my picture here, I have some very small nuclei of these nurse cells. You can see one here, you can see one here, and all of this blue that's surrounding everything, right, that is nurse cells. Notice how my spermatocytes are not directly in contact with the nurse cell. They're kind of separated from it, right? This lumen that's here kind of like branches out and encircles all of these developing spermatocytes. Within the nurse cells is a structure that's known as the blood-testes barrier, and it separates the basal compartment of the nurse cells. This is the basal compartment on the outside. It separates this basal compartment from what is called the luminal or adluminal compartment, where developing sperm cells reside. This barrier, it protects developing sperm from immune system attacks. Remember, the body does not recognize these sperm cells as self cells. They lack identification tags so that they can enter into the female and be undetected as a threat, which they're not. But while it's not a distinct physical line like in the respiratory system where you had upper and lower, this barrier plays a really important role in protecting your spermatogonia. The basal compartment contains your own stem cells, while that luminal compartment contains developing sperm cells, which are antigenically different and could trigger an immune response without the barrier. So this blood-testis barrier, it results from very tight junctions between these nurse cells, and those tight junctions primarily prevent autoantibodies from harming or attacking the sperm.